much. I'm going to share my screen. Can you see well the slides? Okay, super. So hi everyone. As Louise said, I'm Laura. I'm a postdoctoral researcher at Copenhagen University, and I'm very glad to be presenting this talk with Youri, who is a postdoc at the museum in Paris. Together, we will be delving into the potential of using paleoproteomics to study cloth materials, which includes both textiles and animal skins. So after a brief introduction to the topic, we will present two different case studies. The first one, presented by Youri, will focus on the analysis of South American camelid textiles, and the second will be on the analysis of textile and skin garments from ancient Sudan. But I want to start by asking, why should we study garments? Garments can tell us a particularly rich story because they are the result of a very complex, long and costly chaîne opératoire, which begins with the procurement of raw materials, but also because once produced, they accompany us, humans, in both life and death. In that way, garments sit at the crossroad between the exploitation of raw resources, craft technologies, and even societal use. Therefore, we can gain insights into many aspects, such as agricultural practices, animal husbandry, landscape use, or even trade connections. Traditionally, these have been studied with visual or microscopic analysis, but the range of scientific methods that contribute to the study of archaeological cloth has expanded dramatically over the past decades, since the first studies that were conducted in the early 2000s, as you can see in this figure. Today, it includes a wide array of biomolecular tools, of which analysis of ancient proteins has received particular attention for species identification. And however, despite the growth of the field, there are still some limitations, and I will now give the floor to Youri, who will talk a bit more on the specific challenges of applying proteomics to keratin-based materials.
Thank you, Laura. So this is probably an image that most of you will be quite familiar with. So a lot of paleoproteomic research is still often conducted on collagen. And for collagen, we're normally dealing with collagen type 1, for which there are two sequences, alpha 1 and alpha 2, which are different. Unless you're working with fish, of course, there are three different sequences. But for keratin, it's a little bit different. So click. Yeah, thank you. So keratin is slightly different and it's a little bit more complicated. There are different varieties. So you have the acidic type 1 hair keratin, for which there are 8, which you can see on the left. And then you have the basic type 2 hair keratins, for which there are 6, and also can see on the left as well. But on top of that, there are also proteins that are associated with keratin, which have the very highly original name of keratin-associated proteins. And on the right, you can see that for humans, there's a whole lot of variety over there. And combine these keratin and the keratin-associated proteins, all of them together, basically, they have a high potential because there are more proteins that we're dealing with. So if there's just one substitution anywhere, this is of course highly useful for any kind of species identification that you want to undertake. But it also makes it slightly more complicated. Click. Thank you. So here we see the human type 1 hair keratins. You can see this is basically a heat map that you can see over here. You can see keratin 1 to 8, under each other. This all sequences, or part of the sequences at least. And the more blue they are, the more comparable they are. So you can see that there are whole sections that are comparable or even identical. So it means that if you want to use keratin for species identification, you really need to know all these different sequences, because otherwise, if you find any kind of peptides that you're assigned to a particular kind of species, you might actually be wrong, and it might actually be a different kind of keratin, or peptide belonging to a different kind of keratin for another species. You really need to know all these different sequences and to be sure that your species identification is actually correct. Next. That being said, there is still a lot of potential, though this is study that one of my supervisors undertook some time ago. They looked into keratin or textile remains from China. They looked into the keratin of sheep, cattle, and goats, and using principal component analysis, they were able to identify several peptides that were specific to cattle, sheep, and goats. You can see over here in the cluster on the left, and they're really able to assign several of these archaeological textiles to the species level using shotgun proteomics. Next. For my project, which is the paleoproteomic analysis on South American camelids, which I'm undertaking with my supervisors, Elise Dufra and Séverine Zura at the Natural History Museum in Paris, we're now conducting research on 4 more closely related species, the South American camelids. Click. And we're dealing with archaeological texta from the Mochica culture, which date from roughly 100 to 800 CE. As you can see on the map on the right, it's in a coastal area of Peru. It's quite arid, as you can see on the other pictures. But basically, there are these several grave contexts, and for many of these from these contexts, textile remains have been obtained, which they're very colorful and very beautiful, as you can see in the bottom part over here. And these are some of the samples that we're dealing with. Click. But for our species, like I said, they're a little bit more complicated than the cattle, sheep, and goats kind of question, because the 4 species that we're dealing with are the vicuña and the guanaco, the top 2, which are the wild ancestors to the domesticated taxa, which you can see in the bottom part, which are the llama and the alpaca, and they have been domesticated roughly 6,000 years ago. But their domestication process is still kind of unclear and kind of contested. So originally it was really thought that the vicuña was domesticated into alpaca and guanaco was domesticated into a llama. But still, modern DNA research is really contesting this story, and it's still a little bit unclear what's happening, what went on. And additionally, we're dealing with these animals. There's a lot of hybridization going on between all the 4 species, basically, which means that we basically have a whole spectrum of South American camelids that we're dealing with. And this is, of course, very important for our paleoproteomic research as well, as the keratin might have changed through this entire process. Click. So previously, my 2 supervisors have looked into this and looked into specifically modern samples in particular, and they were looking for kind of species-specific marker peptides, using shotgun proteomics, but they were not really too successful. They found one potentially showing up in certain kind of vicuñas, but not in all of them, and many of these others that they found, they were showing up at the whole range of different kind of species. Click. One of the problems that they faced was that many of the sequences for the different kinds of keratin were not known. So you can see here on the left, you see the acidic type 1 and the basic type 2 keratins for the different kind of species. If one of the cells is green, that means the entire sequence has been reconstructed. If it's red, they're not reconstructed, and if they're yellow, then only part of the sequence is reconstructed. So you can see over here that for alpaca, some of the work has already been undertaken. Many of the sequences are already known. But for the llama, vicuña, and guanaco, nothing is really known. And of course, that prevents us from identifying any of these taxa remains to these 3 species. Click. So what we did is then collaborate with bioinformaticians here at the museum. We used publicly available genomic data. We transcribed those into proteomic sequences. So we did this for 2 alpaca genomic assemblies and 2 llama assemblies, 1 vicuña and 1 guanaco assembly. And that basically were 6 assemblies that we're working with, and we've transcribed all of the genomic sequences into proteomic sequences. So it means that per assembly, we have 7.5 million proteins, bringing the total to 45 million proteins, which is, of course, a lot of proteins. But we really want to make sure that we were capturing the keratin and the keratin-associated proteins, as well as any other kind of proteins that we could potentially find in our archaeological material. Click. So what we then subsequently did is we sampled several of the modern known species hair specimens again. We performed shotgun proteomics, LC-MS on these sequences, and really assess what kind of which proteins in these protein databases that we constructed or showed up the most and were potentially useful for species identification. And using blast searches, we were able to reconstruct many of the different sequences for the different kinds of keratins, as well as the keratin-associated proteins. Click. So here was what we previously had. So many of the llama, vicuña, and guanaco sequences were not known. Click. But now we're here, so you can see many of the cells have now suddenly turned green, which is great, which is very happy with. Many of the sequences we've now been able to reconstruct. It means that we could potentially identify our archaeological taxa to the species level. Click. But first, we had to make sure that there were species-specific markers in these particular sequences. So we looked into the different kinds of the species. So we saw that there were, among at least the keratins, excluding the keratin-associated proteins for now, We have two guanaco, two alpaca, six vicuña, and two llama species-specific substitutions within the different kind of sequences. There are more substitutions, as you can see in the right part in the table over there, but many of these were showing up in two or three of the species and were not really species-specific in this kind of way, but are still useful to us, potentially. Click. But one of the problems that still have to face is that, as I said, it has two llama assemblies and two alpaca assemblies. And what we saw is that for certain kind of sequences of the proteins, there were substitutions, or the differences at least, between these two llama and between these two alpaca sequences. You can see part over here. This might have something to do with hybridization going on, or with selective breeding going on, altering the sequences a little bit. So we really have to make sure that we're at the moment, we're not entirely sure which of these sequences we might potentially expect in the past, of course, which might have been different from what they're currently would look like. So this is one of those next steps that we have to undertake and see which of these sequences are actually useful for us in order to identify our archaeological specimen to the species level. Click. And this is where we're currently at. So basically we sampled around 100 archaeological textile specimens. We formed the same method using the reconstructed sequences for the keratin, as well as the keratin-associated proteins, which I haven't showed you here, but we use those as well. And we'll now be able to hopefully assess which of these, whether our work was fruitful, and whether we will be able to identify any of these archaeological textiles to the species level, but we're quite hopeful right now. Click. But basically, this is what we're currently at. So actually, tomorrow I will start this entire process and actually see whether it was successful. But just not in time to actually report back on this during this conference, but hopefully in a month, I actually will be able to tell you all whether it was successful, yes or no. But now we're moving on to Laura's part of the talk.
Thank you. I'm now going to move away from Peru and invite you to travel to Sudan, where I've been working on the identification of animal species that were used for wool and skin production. In general, organic materials are very poorly preserved in the archaeological record, but on rare occasions, like in the Nile Valley, extreme aridity has allowed the exceptional preservation of thousands of textiles and animal skins, as you can see in these examples. So the project Fashioning Sudan will use this tremendous treasure to examine dress practices and past identities as a multifaceted phenomenon. Like this, it will retrace connections between the garments, the natural environment, the economic production, and the social practices. The animal world has tremendously influenced the economy of ancient Sudanese societies, and even today, Sudan is still inhabited by pastoralists, which move across the landscape with their herds. Different production models coexist, such as hunting, nomadic or semi-nomadic pastoralism, and sedentary agriculture. All of these respond to the huge variety of environments that are present in Sudan today and in the past. In the past, the most important livestock in their subsistence were goat, sheep, cattle, and donkeys, but also dromedary, which was a later adoption in the region, since it arrived only from the Arabian Peninsula during the first millennium BCE. And all these domesticates were raised for various purposes, but their use in garment production remains unexplored. They also hunted wild animals, and we know that Nubians were wearing skin and fur garments made of wild exotic animals, as many of them appear represented as such in Egyptian paintings. However, these images were produced by other people outside of Sudan and Nubia, and often are loaded with quite a lot of political and colonial agendas. And it is also very hard to relate these scenes with actual garments. So in this project, we are trying to move away from dietary and symbolic studies and focus on cloth production, which is largely unassessed. Here we will study what animal resources were used to make these garments and examine how garment production affected animal management and economy in general. To do so, I've been using paleoproteomics to examine temporal and geographical changes in the use of sheep, goat, or camel for wool, and domesticated or wild animals for leather and fur. We have selected more than 125 archaeological garments made of wool, leather, or fur. The leather has been analyzed using zooms and keratin materials with shotgun proteomics. We have also collected modern hair references for Sudanese sheep, goat, dromedary, giraffe, or gazelle to obtain full keratin sequences for those species that are not present in current databases or that have incomplete sequences. In total, these comprise more than 15 archaeological sites, spanning from prehistory to the medieval period, and from a very large territory stretching from Lower Nubia to central Sudan. So moving on to the results, for leather, we tested 35 samples, but identification at the species level was not possible. This could be due to either archaeological degradation or manufacturing techniques. I personally think that it must be related to the application of colorants like red ochre or other treatments that might interfere with zoom results. We've had positive results from Egyptian leather, which present very similar preservation conditions, but were maybe manufactured differently. So this will require further investigation in the future. In terms of the wool remains, I am still working on the data analysis, so I'm just presenting some preliminary results that show basically that the majority of samples were sheep, but we have a few cases where it could be potentially dromedary, and surprisingly, we didn't find any goat hair so far. Finally, I would like to highlight another aspect of the project, which is to develop case studies of well-preserved clothing assemblages and to see what they can tell us about the identities of the person wearing them. Here is a mummy from the medieval site of Eterev that is currently preserved at the British Museum. A sample from this multicolored wool blanket that was wrapping the individual was identified as dromedary. And I think this raises interesting questions about the development of camel-based pastoralism and desert networks. But also we can look at questions of dress practices, social status, or diversity of funerary practices in Christian Nubia. And finally, what interrelations happen between different cultural groups in the region. So to conclude, we hope to have shown you the potential of using proteomics in cloth materials with two very different case studies. At the same time, Youri and I wanted to end this presentation by highlighting some of the current limitations. The most important is the need to reconstruct full keratin sequences for species of interest. In addition, for keratins, there are many repetitive sequences that are shared among many species. On top of this, there is a lot of interspecies variation. So all of this renders identification very difficult at the moment. Therefore, there is still a lot of work to do to assess the full potential of paleoproteomics applied to cloth materials. And with that, I would like to thank you all for your attention, and we are both very happy to take questions.
Can you see the screen? In a minute, maybe? Yeah.
Yeah,
Perfect.
looks good.
Okay.
Right. I hope I won't have any issues with internet connection or anything today. So let's do our best. So hi everyone, and also greetings to all the invisible audience over there on Zoom online. And we're going to present to you with Katarina just shortly how and why it's actually good to venture out sometimes from the safety of MaxQuant and to explore different software and their potential.